In 1824, Marie-Jean-Pierre Flourens detailed ablation to study behavior. Flourens was a French physician, famous for various significant discoveries relating to the nervous system, cerebral localization, and brain functions. He pioneered the experimental method of carrying out localized lesions of the brain and observing their effects on movements, sensibility, and behavior. By removing anatomically defined areas of the brain of an animal and watching its behavior, he thought he might localize certain functions. He used ablation and stimulation on mammalian species, specifically rabbits and pigeons. Previous researchers had lesioned the brain through a trephine, but with that technique it was impossible to localize damage accurately. Flourens completely uncovered and isolated that portion of the brain to be removed. He tried to minimize operative trauma and post-operative bleeding and infection, which might have confounded analysis. Flourens was able to demonstrate convincingly for the first time that the main divisions of the brain were indeed responsible for largely different functions. By removing the cerebral hemispheres, for instance, all perceptions, motor functions, and judgment were abolished. The removal of the cerebellum affected the animal's equilibrium and motor coordination, while the destruction of the brainstem caused death. These experiments led Flourens to the conclusion that the cerebral hemispheres are responsible for higher cognitive functions, that the cerebellum regulates and integrates movements, and that the medulla controls vital functions, such as circulation, respiration, and general bodily stability. However, he was unable to find specific regions for memory and cognition. This was probably due to the fact that his experimental subjects had fairly primitive cortices. Flourens' initial intention was to investigate localizationism. In other words, he aimed to understand whether different parts of the brain had different functions, as the Austrian physician Franz Joseph Gall was proposing. Flourens was also a strong critic of phrenology, which was developed by Gall. It stated that the brain consists of independent functioning units. These areas were considered to be responsible for different intellectual aptitudes and characteristic traits, and how the skull bone reflected these differences. However, the idea that personality is determined by skull shape is now understood to be incorrect. According to Flourens' theory, the brain acts only as a functional entity, although specific functions are controlled by specific parts of the brain. This concept of cerebral equipotentiality can be observed in his book, entitled Experimental Researches on the Properties and Functions of the Nervous System in Vertebrate Animals. Flourens accepted that the cerebral cortex, cerebellum, and brainstem function globally as a whole, equipotential, and in conjunction with every other part. He found that all cognitive functions were interrupted in pigeons when the cerebral hemispheres had been taken away. Flourens did not favor the idea of cerebral localization and concluded that the brain functioned as a whole when he wrote, "A large section of the cerebral lobes can be removed without loss of function. As more is removed, all functions weaken and gradually disappear. Thus the cerebral lobes operate in unison for the full exercise of their functions. The cerebral cortex functions as an indivisible whole, housing an essentially single faculty of perception, judgment, and will, the last refuge of the soul." Flourens made other contributions to science as well. In 1825, he observed that when a pigeon's horizontal semicircular canal was destroyed, the bird continued to turn in a circle, thus demonstrating the function of this vestibular labyrinth. He thus proposed that the semicircular canal was involved in the maintenance of posture and balance. And in 1874, he described the anesthetic properties of chloroform and ether.